Hello grade 7 students. In this video we're going to talk about the exercises about self and the non-self. So let's start with the self, the biological self. Grafts, transplants and blood transfusion are common medical practices. They can save the life of many people, but in order for one of these techniques to be successful, certain conditions must be ensured. So what are these conditions? Let's start first with self markers, the HLA system. The markers of the immune system they revealed by grafting are molecules located on the membrane. They are known as HLA, human leukocyte antigen. They are proteins. They represent the immunological identity card of the individual. So they are like the ID for the individual, and they are unique for each individual. So they are unique, meaning that they are special. So every individual has a special HLA, a unique HLA. Nowadays, the term MHC, major histocompatibility complex, is used to define the tissue compatibility, how they are similar between the donor and the recipient. So HLA is also called MHC, major histocompatibility complex, to see the similarity between the cells, between the donor and the recipient. As we know before, we have 4 different types of graft. They are called autograft, that is done between the same individual, and always the result is tolerance, meaning that the graft is accepted, like this case. The graft here is accepted. Isograft is done iso, identical, between identical twins, and the result is tolerance, meaning that the graft is accepted. So this is the case. Allograft, different individuals belonging to the same species, between me, for example, and Hassan, between Zahra and Zainab, and so on. So the result, in normal case, the result is rejection and will cause this redness. This is the rejection case. Also, the 4th type is called xenograft, individuals with different species, and the result is rejection. The graft is rejected, it's not accepted. So solving probably the activity, pick up from paragraph 1. We're going to solve it after finishing document C. Document C, degree of kinship between individuals and the percentage of the graft survival. What is the percentage of the graft survival? If they were identical twins, the percentage of survival is 100. So it's maximum when they are identical twins. Brothers and sisters, it decreases to 80. And if they are unrelated, they don't relate to each other, meaning that, for example, they are friends, let's say, or they are neighbors, this percentage decreases to less than 40%. So here, pick out from paragraph 1, part A, what differentiates the 4 types of graft. Here, the four types of graft, they are different in the donor and the recipient. In the auto, the graft is done within the same individual. Iso, identical twin. Allo is done between different individuals belonging to the same species. Xenograft, different individuals and different species. So the 4 types of graft, auto, iso, allo, and xeno, they differ in the donor and the recipient. Part B, the cases where the graft is tolerated and where it's rejected. So the graft is tolerated where? In the autograft and in the iso, and it's rejected in the case of allograft and xenograft. Part C, what are the markers that define the tissue compatibility between the donor and the recipient? They are called MHC, major histocompatibility. Compatible, they are similar. So major histocompatibility complex, or it has another name, it's called the HLA. Okay, so let's interpret the result of document C. Interpret what the meaning of this, analyze plus indication or the significance. So the percentage of the graft survival is 100% when the graft is done between identical twins. Here, doing analysis. I don't have to say greater than or less than. We have to say increase or decrease. This percentage becomes 80, so it decreases. So this percentage decreases to 80%, where in the cases of brothers and sisters. And it also decreases to less than 40% in the case of unrelated individuals. What's the meaning of this? This means that, or this indicates that the graph is accepted without any risk, is accepted 100%, where in the case of identical twin. Okay? Part 3. Formulate a hypothesis explaining the high percentage of graft survival in the case of identical twins. What can we say about this hypothesis? We can say that hypothesis, maybe the identical twins, they have identical HLA, or they have the same HLA. Okay? Part 4, they say specify the percentage of graft survival between non-identical twins. Non-identical means that they are brothers and sisters, so the percentage is 100 because they come from 2 fertilized eggs. They are called brothers and sisters, and they have the same percentage of success. Now let's move to other markers of the self. Blood groups. Blood groups, you know that we have 4 different types of blood groups. We have blood group A, blood group B, blood group AB, and blood group O. Here in the blood group, we have some markers. We have something called antigen, and we have something called antibody. So from now on, you have to know this idea. Antigen is a molecule foreign to the self and can trigger the immune reaction. It's found in non-self and can cause a reaction. Antibody, it's formed against the antigen. For example, we have antigen A. Let's start with the first group, group A. Group A, always the blood group has the same antigen as the blood group. We have antigens A and antigen B, and we don't have antigen AB or antigen O. So blood group A, it has antigen A. So blood group A, it has antigen A. Blood group B, it has antigen B. As we notice here, the blood group and the antigen, they are the same. Blood group A, antigen A. Blood group B, antigen B. Blood group AB, they don't have antigen AB specific. They have antigen A and antigen B. So you have 2 antigens, A and antigen B. And there is no antigen AB, one antigen AB. We have antigen A and antigen B. While the surprise here that blood group O has no antigens, since we don't have antigen O. Only we have antigens A and antigen B. Moving to the antibody. Antibody is something that can attack the antigen. So antigen A, meaning that we have blood group A. So antibody should be different from antigen A. So we should have antibody B. So as a conclusion, the blood group has the same antigen but different antibody. So antibody is a molecule that is released against a given antigen. For example, my blood group is A. So I have antigen A. What type of antibodies? I have antibody different from A, which is antibody B. Moving to the second blood group. Blood group B. Blood group B has antigen the same as the blood group. So we have antigen B. But antibody, it should not be the same as the blood group. So we have blood group B, it has the same antigen, antigen B. But the antibody should be different. So antibody should not be antibody B. So it should be antibody A. Why? We will notice this after a few minutes. So why we should not have the same antigen and antibody in the same blood group. So if my blood group is A, I have antigen similar to the blood group, so I have antigen A, but antibody should be different. If you have antigen A, the antibody should be antibody B. Second case, if I have blood group B, so the antigen should be similar to the blood group. I have antigen B, but the antibody should be different. So it should be antibody A. And blood group AB, it has antigen A and antigen B. So we have two antigens, A and B. So what type of antibody? The surprise here, we don't have antibody. Since if we have anti-A, it's going to attack the antigen A. If you had antibody B, it's going to attack antigen B. And this will cause a real problem. We're going to talk about this later. Blood group O, there is no antigens. So we can have any type of antibody. So the antibody is antibody A and antibody B. Both of them, they are present. So ABO system, the blood group is called ABO system, or the blood group system is characterized by the presence or absence of molecules on the membrane of the erythrocytes, known as markers A and or B. They are named agglutinogens or are called antigens. So antigens and agglutinogens, they are the same. They are present since birth and they were identified by Landsteiner in 1900. Antibodies or agglutinins. So whenever you hear the word agglutinin, meaning that antibody. Anti-A and or anti-B, they are present in the blood group and the blood plasma, where the corresponding agglutinogens are present in the red blood cells. So you have to know this note. Identification of the blood group. How can we identify the blood group? Identification of the blood group is somehow a complex process. It needs antigens and antibody. So here we took a blood sample of an individual. For example, Ahmed enters the lab. He asks, I want to know my blood type. So what they will do, they will take drops of blood, 3 drops from your finger. So 3 drops are going to place on a slide, and then these 3 drops, they are going to be subjected for anti-A, antibody A. We said that antibody A will attack which antigen? The same, antigen A. Antibody B will attack antigen B. And the last one is for confirmation. So here we have, here the blood group, it remains the same. But here we have the blood group, they look like they are, for example, destroyed. This destruction is called agglutination. What's the meaning of this? Agglutination meaning that when the antigen and the antibodies, they meet together. For example, anti-A, it will meet with which antigen? Similar. So it will meet with antigen A. So this meeting between antibody A and antigen A is called agglutination. By this way, we can know that the blood group here is blood group A. Now let's move to the second blood group B. We said that if we have blood group B, meaning that we have the same antigen, we have antigen B. We can write the antigen as AG, antigen B. So after adding anti-A, there is no antigen A. So there is no agglutination. The blood sample become intact, become the same. It stays the same. While here, when we add anti-B will attack the antigen B. So we have agglutination. So here we have agglutination. So meaning that we know that this is the blood group, is blood group B. So here, we don't know in the lab, we don't know that we have blood group B. So after adding anti-B, agglutination, meaning that the antigen and antibody, they mix. So as a simple definition for the agglutination, agglutination is the for example, the meeting of the antigen and its corresponding antibody. What the meaning? Meaning this means that whenever we have the antigen and its corresponding antibody, for example, we have antigen A. What is its corresponding antibody? Is antibody A. Antigen B, antibody B. So anti-B when it's added, it makes agglutination here. So we know that anti-B, it's find its corresponding antigen, which is antigen B. And by this way, they can know that this is the blood group. Blood group AB. What do we have? We have antigen A and antigen B. So I write AG is antigen A, antigen B. So here we have agglutination. So anti-A makes agglutination, so we have antigen A. Anti-B makes agglutination, we have antigen B. So we have antigen A and antigen B together. So the blood group is going to be AB. Anti-A, there is no agglutination here. So there is no antigen A. Anti-B, there is no agglutination. So there is no antigen B. So no antigen A, no antigen B. Which one of this? It's a blood group O. So we determine the blood group by mixing a drop of blood group with a drop of serum. That serum, it contains the antibodies. Here the serum, it contains the antibodies. If the recipient doesn't have antibodies against the agglutinogens of the red blood cells of the donor, transfusion is possible. So we can have transfusion. And no agglutination is provoked. It's said that there is a blood compatibility between the donor and the recipient. Part 5, they said draw a table that shows agglutination and agglutinogens. We're going to draw this. Part 6, indicate why the blood of an individual must not contain at once the agglutinogen and its corresponding agglutinin. Already we discovered this. Why the blood group of an individual should not contain the agglutinogen, meaning that the antigen, and the agglutinin, the antibody at the same time. Why? Because we will have agglutination and the red blood cells are going to be destroyed. So agglutination and agglutinin should not be present at the same time with the same type. So there is no antigen A and antibody A at the same time unless we're going to have agglutination. They said that blood group O is called a universal donor. Why? Blood group O, if you see here in document D, blood group O contains no agglutinogen. There is no antigens. So when blood is transfused into another blood group, we have to look at the recipient, we have to look to the antibodies. So if you have antibody A, there is no antigen A. If you have antibody B, there is no antigen B. So it's called the universal donor. So blood group O individual can donate to all blood groups. While blood group AB, it contains no antibodies. So it's called a universal recipient. Move to the answers. How do the different blood group, different types of graft differ? The 4 types of graft, they differ in the donor and the recipient. The graft is tolerated where? In the case of autograft and isograft, but it is rejected in the case of allograft and xenograft. The markers that define the tissue compatibility between the donor and the recipient are called the HLA, also called MHC. MHC major histocompatibility. The percentage of the graft survival is 100 when the graft is done between 2 identical twins. This percentage decreases to 80% when the graft is between brothers and sisters, and also decreases to less than 40% when the performed graft is done between 2 different unrelated individuals. So what does this indicate? This indicates that the transplant is accepted without any risk. It's 100% accepted in the case of identical twins. Did you notice here that we have analyze? This is the indication in blue. So we have analyze. In analysis, you see here, I said decreases. Decreases. So you have to say decrease and increase. Okay? Hypothesis, maybe the identical twins, they have HLA molecules. Moving to part 4, the percentage of graft success among non-identical twins is 80. Why? Because they come from 2 different fertilized eggs. They are brother and sister. They have the same percentage of success. Table. So blood group A, as I said, they have the same agglutinogen. Agglutinogen, I said, it's similar to antigen. And agglutinins is the same as antibody. So here, blood group A, we said it has antigen similar to the blood group. So it has antigen A. And antibody different from A. It's going to be B. Blood group B, it has the same antigen. So blood group B has antigen B, while it has antibody different from B. So it's going to be A. So always the antibody is opposite. So always here, let's see here, B and A, they are opposite, while antigens, they are the same. So always the antigens are similar to the blood group, while antibodies, they are the opposite. Blood group AB, so we have antigens A and B, while we don't have any antibody. So there is no antibody here, no antibodies. Antibody, we can write it as AB, antibodies with small s here. So no antibodies. That's why this blood group AB is called the universal recipient. Why? Because in the blood transfusion, whenever you have to have blood transfusion, for example, and certain individual need blood. So this is called the, for example, the transfusion of blood. So transfusion of blood here, blood group AB, it can take blood from and blood group A, from blood group B, and from blood group AB. Always the donor, we have to look to the antigen. Pay attention to this note. Always in the blood transfusion, the donor, the one that donates or gives, we have to look to the antigen. And always the recipient, the one that receives or takes, we have to look to the antibodies. So here, blood group AB, no antibodies. So there is no possibility to react with antigen A or antigen B or antigen AB. So blood group AB, it can take blood from blood group A, from blood group B, from blood group AB for sure, because the same blood group, and also from blood group O. That's why AB is called universal recipient. So it can take blood from different blood groups since there is no antibodies. Moving to the blood group O. Blood group O, there is no antigens. No antigens. Why? It has antibody A and antibody B. So antigens, always you have to look to the donor. So donor has no antigens. So here, blood group A and blood group B. We said that blood group, blood transfusion, we have to look to the antigens in the donor and to the antibodies in the recipient. So no antigens. In the recipient, blood group A has antibody B, so no antigen, so it can work. Blood group B, it has antibody A, so no antigen, so it can work. Blood group AB, it has no antibodies, so it works. And O for O, and here it works. So here we call blood group O universal donor. It can donate for all blood groups. So blood group O, هو كريم, it can donate blood group to all blood groups, while AB بخيل, it can receive from all blood groups. It's called universal recipient. Okay. So here in part 6, they said the blood of individuals should not contain simultaneously, meaning that at the same time, the agglutinogen, meaning that the antigen and the antibody at the same time. Why? Because if we have, let's say, antigen A, the blood group A, antigen A, and we have the same antibody, antibody A. so they will attack each other. So antibody A will attack the antigen A and agglutination will occur. That's why the antigen and its corresponding antibody should not occur. Part 7, blood group O, as we see before, doesn't contain any agglutination. There is no antigens. So on the membrane of its erythrocyte we said it's red blood cells. So when blood is transfused into another group, the agglutinins, the antibodies, will not find any enemy. There is no antigens, and antibodies should fight against the antigen. So there is no agglutin. The agglutinins couldn't find anything to agglutinate with, to fight with. That's why it's called universal donor. So it can donate blood to all different blood groups. Besides meaning red blood cells. Moving to group AB. Group AB is characterized by the absence of agglutinins A and B, and by the absence of agglutinins. So they can receive blood from any other group, and it has no agglutinins to agglutinate, to make agglutination. So it's called universal recipient, as we said. So hope this video help you to understand well about the blood groups and to know more how to solve exercises related to the grafting and related to the blood transfusion. Thank you guys. Bye bye.